for the first time, we can actually say that, your genetics is not your destiny, right? Like you really, no matter what luck of the draw you got when you were born, you don't have to suffer from that for the rest of your life. And I think that very much could be, a major triumph of human engineering.
Welcome to Unnatural Selection, a show about competition, innovation, and the strategies shaping tomorrow's frontiers. I'm Nick Encina. Each week, we speak with pioneers shaping the future.
Episode 30 of Unnatural Selection, produced in partnership with Mass General Brigham, ahead of the World Medical Innovation Forum. In this episode, we speak with Trevor Martin from Mammoth Biosciences about the revolutionary potential of CRISPR technologies in treating disease. We dive into the remarkable story of baby KJ, the first newborn to receive a personalized CRISPR therapy for a rare metabolic disorder, and explore what this breakthrough could mean for families affected by rare diseases. We also look beyond rare conditions to the promise of CRISPR in tackling common diseases like cardiac disorders, with the potential to save thousands, even millions of lives. Dr. Trevor Martin is the co-founder and CEO of Mammoth Biosciences, a biotechnology company based in the San Francisco Bay Area that is harnessing the diversity of nature to power the next generation of curative CRISPR therapies for genetic diseases, as well as improving human health and well-being. Through its AI-enabled discovery and development of novel CRISPR systems, including from co-founder and Nobel Prize winner Jennifer Doudna's lab, Mammoth is enabling the full potential of its platform to read and write the code of life. His scientific work has been featured in outlets including 538 and The Atlantic. He is the featured healthcare honoree on the Forbes 30 Under 30 list, is on Fortune's 40 Under 40 list, and is an EY Entrepreneur of the Year. Let's go.
Trevor, welcome to Unnatural Selection.
Yeah, thanks for having me.
To level set and just give everybody listening some context around what motivates you, could you please tell us what need or impact drives your work?
Yeah, I think that Mammoth is part of two revolutions in biology that are very exciting. The first one is more on the research side, and that's this idea of biology becoming more like engineering. And instead of being this kind of like adventure into the dark jungle and trying to build a drug by kind of feeling around in the dark, how can we build reproducible systems that don't just build one drug, but can build hundreds of drugs where it gets easier every single time? And then the second one is more clinical, and that is how do we move from treating the symptoms of a disease to actually curing it fundamentally and doing things like one-time curative therapies. So I think both of those are really exciting, and when you combine them together, it's even more compelling. And that's really the story of Mammoth, and I think the story of CRISPR more generally is how do we create reproducible, programmable medicines that actually cure disease.
I love it. And I have a software background as well, so it's almost like you're building an operating system for drug discovery using CRISPR technologies.
Yeah, exactly. And I think that's been kind of the holy grail for a long time is like, how do we change biology from this kind of squishy, messy, like bespoke, handcrafted, artesian discipline into something that is frankly more boring and formulaic and more of like a factory. And I think there's a lot of different components that come together to make that happen. And I think that kind of goes into broader trends. People love to talk about kind of AI and drug discovery and things like that. And these are all other elements that come together to make drug discovery and development more formulaic. But I think really underpinning all of that, the thing we're obviously most excited about is this idea that CRISPR is this molecule that means you don't need to go and rediscover a bunch of fundamental biology or anything for any genetic disease. You can really reprogram the same molecule very quickly, very effectively to go after thousands of different diseases and kind of know that it's going to work.
Hmm. In CRISPR has become a household term in science conversations, but its applications are incredibly diverse. Where is Mammoth focusing its bets and why those areas, say, over others?
Yeah, so we're focused on therapeutics and building these curative therapies for genetic diseases. To your point, though, CRISPR really has applications anywhere where you want to edit DNA, and DNA is or RNA for that matter, and DNA is the code of life. So that has very broad applications, anything from biomanufacturing. So how do you build some new material that doesn't exist in nature, all the way through to, of course, therapies. How do you edit someone's DNA to correct a mistake that's causing a disease, and many things in between. For example, I think many people know us for the work we've done during the pandemic on the diagnostic side, building a COVID test using CRISPR. There's also exciting applications in research in terms of like how do you screen for drug targets by doing massive editing of different like cell lines so you can knock out all sorts of different genes and even combinations of genes and see what the effects are. So it's really just kind of this new think of it as like when the microscope was invented or when PCR was invented, but even more applications because it actually can be a diagnostic, can be a therapy. It's not just a kind of research technology. And I think underlying all of that is this, again, fundamental idea that it's this programmable technology. Because one interesting thing to note is that CRISPR is not like the first time we've been able to change DNA. There's other technologies like restriction enzymes have been able to change DNA technically for decades, but could only go to one location. And you just had to hope that maybe that location was useful. Zinc fingers have come before. There's all sorts of kind of proteins that can bind to DNA and RNA in various ways. I think one of the main reasons that CRISPR has taken off in all these different fields in the way that it has is really because of that reproducibility across different targets and really that ability to truly program it very easily without having to kind of reinvent the technology every single time you're going after a different target, whether that's research or clinical development.
And since we're on the topic of research and clinical development, CRISPR is obviously a technology that's evolving incredibly fast. And when you think about innovation in biotech, what do you see as the difference between kind of like novel science and usable innovation? So, in other words, how do you decide when an idea is ready to move from the lab bench to real world impact?
Yeah, that's a great question. And I think not enough companies ask themselves that question early on. I think we've tried to be very diligent about always asking ourselves that question. Because, look, I mean, as a former PhD scientist, I love novel research. And I think there's many, many reasons why that's beneficial to society in a variety of ways. But when you're founding a company, the key step I think between novel research that's like really cool academically and adding to the body of knowledge and you don't know necessarily where it's headed over decades and something that should turn into a company now is really okay, do forget technical risk and all these other things. There's always a lot of technical risk, but assume everything works out. Take your technology. Will you be able to do something in the world that is not possible with other technologies? Because I think often scientists do get too wrapped up in like, and especially in the CRISPR space, let's be honest, like, oh, this is like cool new thing. And like, oh my God, such a cool mechanism. Okay, cool. At the end of the day, are you going to be able to build a drug, let's say, that's going to do something either more effectively or that would not be possible, period, with any other technology that's out there? Ideally, a weaker version of that would be could you do it more cost effectively or quicker or something? Those are fine reasons, but definitely weaker, I would say, than can you do something that's not possible or can you do something more effectively than anything else? And if the answer to that is yes, yeah, sure, found a company. I think that's amazing. And if the answer to that's no, either keep looking and try and figure it out, or maybe it's not right for a company and it's an amazing research paper. And I think that's something where, especially when there's a lot of cash floating around in the biotech ecosystem, it's hard to imagine that today, but 3, 4 years ago, that was the case. Um, I don't think many people necessarily ask themselves that question. So you get all these amazing technologies that are super cool academically, but then it's like, okay, and this is what we're going to use it for, and it's like there's 5 other drugs that are doing that exact thing. So I think that that's the key question that you have to ask yourself, in my mind, is like, what am I bringing into the world that's not possible otherwise? Because the patient doesn't care what the technology is. You have to assume that.
Yeah, exactly. What the mechanisms of action are irrelevant to the patient as long as it cures, treats, or makes some kind of illness more manageable for them.
Yeah, exactly. So I think even asking yourself that question, there's still just infinite really cool companies that can be founded. But I think it's good to focus on companies that can very clearly answer that question. Even if there's a ton of technical risk, that's fine. Like everything has to work right for this to happen. That's cool. As long as you can answer the question at the end of the day of why that risk is worth taking. Yeah.
Yeah. And obviously you work in the DNA space, and I'm also in that space in the genomic newborn screening world. And so I know that people tend to be very sensitive and sometimes even maybe um, I don't want to say paranoid, but protective of their DNA, right? And so, there are regulations. We have to account for public trust, and that can make or break biotech breakthroughs. So, how do you see the sensitivity of people around DNA? And of course, that's a little bit less of an issue when you're dealing with some horrific disease, right? Because in that situation, people are pretty willing to overlook privacy or any number of things because you're dealing with something much more immediate and much more critical. But nonetheless, we're still dealing with DNA. So, what's the biggest external constraint and innovation you face today when it comes to working with people's DNA?
Yeah, well, I think that's why you're seeing most genetic medicine starting with extreme needs where it literally is a life or death situation for the patient, and maybe a genetic therapy is the only way that they will survive. Makes sense, of course, to start with things like that as the safety is being proven out, and for any new technology, you want to always find that right balance between the risks and like kind of the unknown versus the benefit to the patient. I think what's exciting right now in the CRISPR and gene editing space and kind of genetic medicine more generally is that we have so much data now across different modalities, including CRISPR, like years of following the first patients that have been dosed in vivo, and of course, even longer for ex vivo work. And what we're seeing is that actually so far, it looks like, all the cards are coming up green in terms of incredibly safe and effective technologies. So I think the next stage that's really exciting, and of course, like you're always, raising the bar for safety and all these other things as you continue to get more data, you can always have more, right? But you're always making that risk reward calculation as we get more and more data showing that the risk is relatively low as far as we can tell. The potential benefits to the patients kind of widen in terms of what's the therapeutic aperture that you could tackle. And I think that's a really exciting moment for CRISPR and gene editing in particular, as we think about moving from more rare to more common disease. Because especially when we think about how CRISPR works, a one-time treatment, right? Like a
Hmm.
single time. It's a very different way of thinking about medicine. You think about like GLP-1s and all these medicines with amazing benefit to patients, right? Like miracle drugs, but you got to take them the rest of your life. And look, pharma doesn't have the best reputation generally. Like, oh yeah, they get you hooked on the drug and then make you pay the rest of your life. Well, genetic medicine is the opposite of that. It's a way of really saying, okay, if we think this is actually a really compelling, efficacious way of addressing something, we can actually one time treat the patient, and then for the rest of their life, they will get that benefit. And I think that that kind of meets the moment as well in terms of like rising healthcare costs, in terms of things like access. So I think we're at this very interesting moment where actually the time is very ripe, in my opinion, for genetic medicines to start moving into some of these more common indications as we get that, growing body of safety data. We have these increasing pressures on the healthcare system. Just every single year, healthcare costs are going up. And I think it creates this very unique opportunity where if the science is very transparent, and I think one of the things I learned from the pandemic is like I think transparency is always valued. Never just try and give like a guidance because that's you're trying to like play some guessing game of like, oh, if I tell people not to wear masks, then there'll be more masks for the surgeon. If I tell them to wear masks, then like they'll make sure to preserve masks that are available for like first responders. Just like don't be too cute. Just explain the technology, explain why you're doing what you're doing. And I think there is an uphill battle generally in terms of kind of public trust of scientific institutions. There's no way around that. And I think in general, the only way through that is going to be very transparent about, hey, here's the safety data for how this works. Here's the benefits. And the whole point of the FDA, which in turn is, governed by obviously our elected representatives, is to find the balance of risk and reward that we're comfortable with as a society. And I think all we can do as companies and scientists is really be transparent about what we see as the risks and the benefits, and then let people decide where they want to stand on that spectrum. And there's
Yeah.
benefits in terms of cost and healthcare access and all these other things, but that's for society and the FDA to weigh at the end of the day.
Yeah, and I think we've seen some extraordinary cases of successes in CRISPR. But just for the sake of just transparency, since you mentioned that, what are the kind of risks that still linger in CRISPR technology that you're focusing on the most right now?
Yeah, I think that in general, what's been really exciting is that I think initially, so the way CRISPR works is that it forms for kind of classic gene editing with something like Cas9. We use novel new systems, things like nanocast and Cas-V. But the way any kind of CRISPR system fundamentally works for what we call a knockout of a gene, where we're kind of turning a gene off, is that it makes a double strand break in the DNA, and through the repair of that break in the body or ex vivo in a cell, it basically introduces a small error, and then that turns the gene off functionally. So the initial thought here was, is there some kind of downstream bad effect of causing a double strand break? That was like just a general question. And you can come up with reasonable hypotheses about why that may or may not be the case. But at the end of the day, the proof is in the pudding in terms of there are now hundreds of patients that have been treated, if not cured, with this type of technology and this type of mechanism. And there don't seem to be any adverse events related to that type of technology. So we'll continue, of course, as a community to follow those patients. And you can always say, okay, maybe 5 years, 10 years, 15 years. But every year that you get additional data, there's less and less ways you can imagine that this could be causing any sorts of problems. I think in terms of what we focus on more, it's more when we're building a therapy. In addition to what we call the on-target editing that you get, so that's the edit that's the preferred type that you're trying to create. You're trying to knock out a specific gene, and you're getting that double strand break that knocks out that specific gene. You can also get what are called off-target edits. So that's where that same CRISPR protein goes somewhere else in the genome and makes a double strand break or kind of any kind of interaction. And that's something where every time you build like a new targeting, what we call RNA, a guide RNA for a CRISPR protein, you really, one of the main considerations is how much on target are you getting versus any off target. And ideally, you have zero. Sometimes maybe there's a little bit, but you're saying, okay, is this in a region of the genome that has some important function? It's like an oncogene or something, or is it in some part of the genome that doesn't have any known function? And people seem to have deletions here with no problems, or ideally, there's just no off target. So that's I think where frankly most of the field is more focused in terms of like kind of day-to-day safety considerations. And that's something where thankfully like we can design many guide RNAs very easily, and that's something where we can kind of screen when we're developing the drug for drugs that either don't have off-target effects that are measurable to any degree, or if they do, it's in areas that we know aren't going to have any kind of like functional effect.
Hmm. In February of 2025, so just this year, baby KJ received a personalized CRISPR therapy for CPS1 deficiency. It's a severe metabolic disorder. Cases like this highlight the potential of genomic editing for rare diseases. And how do you see these individualized therapies evolving as genomic newborn screening becomes more widespread? And what challenges need to be overcome for this to become a more common reality for families?
Yeah, no, that's super exciting. I think a demonstration of the power of editing as well, right? Like this is something where it would be difficult, if not impossible, to that quickly come up with some drug that treats the symptoms of the disease. But because of the screening that was done to catch what was happening, and because of the availability of technologies like CRISPR, we're able to very rapidly actually come up with not a treatment, but potentially a cure for what was going on. So I think some of the remaining hurdles there are, thankfully in many ways kind of practical ones. I think one is regulatory. So of course, how do you make sure that patients that want access to this type of technology can get access to it? And it seems like there's a lot of excitement about helping with that. So that's super encouraging. I think the second one that's practical is manufacturing. A big part of building any drug is kind of the less seen part, which is how do you actually get what's needed into a syringe and make that as safe as absolutely possible so that you don't have any complication just because of the manufacturing process of that drug. And especially when you're manufacturing for a single patient, for example, that can be very difficult to scale. And it relates to the third practical one, which is the incentives around this and how do we enable this to have a viable business model, basically. And it goes a lot towards the what we call CMC, the manufacturing. And I think that there's a lot of kind of remaining questions, I would say, in terms of like, okay, we solve all the regulatory problems. Let's say we can figure out how to get the CMC costs low without sacrificing any sort of like safety issues. okay, this is a single patient. What are we going to charge? $5 million, $10 million? Even then, is that a sustainable business? If
Hmm.
depending on the rest of the cost structure around it. You also don't know like how many patients are necessarily going to be opting into this. So it's just very tricky to really build a kind of more scalable version of it. I think that's the big question on the table. I think it's super exciting that we now have these proof of concepts that we can do this. And I hope this will continue to happen and continue to show the power of this technology. But I think the really interesting nut to crack there is how do you create something where companies like us can really help out with these types of initiatives in a way that's sustainable long term and is not charity because charity is great but it's not sustainable, and it would be awesome if
Yeah,
this is something where companies could be extremely incentivized to help as many people as possible.
no, it makes total sense. Um, what does the innovation landscape look like right now for CRISPR? What are the things that excite you the most about this application in the near future?
Yeah, so I think in general, one of the things that really excites me about CRISPR and kind of genetic medicine space in general is definitely moving from more rare to more common disease. I think that's super exciting and has the potential to help not just tens of thousands of people, but millions of people. Like, can you imagine basically getting a vaccine against like cardiometabolic diseases, right? How many millions of lives could you save if you lowered the number of heart attacks in the US by 50%, right?
Yeah.
So I think that those lines of inquiry, how much money could you save the healthcare system if you could do that? So I think those lines of inquiry are becoming increasingly real and very exciting. I think also one of the things that we've really helped pioneer at Mammoth as well is how do we actually get to every cell in the body, right? I think that there has been so much amazing work ex vivo. There's so much amazing work going on from us and others in the liver. Well, if you think about genetic disease, what do you think of? Alzheimer's, Parkinson's, Huntington's, right? These are all like CNS, muscle diseases. And it's been really, really tough to figure out how to get these technologies to those tissues. So, one of the things we're really excited about at Mammoth is that we have these really tiny versions of CRISPR that can actually fit into some of the delivery mechanisms that we already know can go to those tissues. So, instead of kind of waiting around for magical new delivery to come around, can we use the delivery technologies of today to deliver the kind of cargoes of tomorrow, like these CRISPR editing technologies, these precision changes to the genome where you're not just turning off a gene, but you're writing in the exact sequence that is healthy, or you're kind of tuning the expression of a gene by turning it down or even turning it up. These are all what we call CRISPR plus methods. You might have heard of it as like base editing or epigenetic editing or gene writing. And at Mammoth, we think that we have line of sight for the first time to actually getting these technologies to these tissues that have these extraordinarily debilitating diseases, and where it's kind of frankly needed most, and just moving beyond the liver and moving beyond ex vivo. I think often of like as an example, like Huntington's disease. This was one of the first genetic diseases ever mapped. I think before there was even the sequencing gels, I think they used microsatellites to map it. And that's, probably almost 40 plus years ago. Yet still today, patients die of Huntington's, right? 40, well, many decades, let's say.
Yeah.
I think genetic medicine will have truly kind of arrived to its full potential when the last patient dies of Huntington's disease.
Hmm.
That has not happened yet. So I think in general, our goal at Mammoth for sure is how do we do any type of edit in any cell of the body, right? And that's how you're really going to be able to deliver on the true promise of genetic medicine.
Yeah, that's amazing. If we, thinking about you sit in a very special and unique place and as pioneering this amazing technology. And if we go full circle to where we started, we talked about you helping to build this platform, if you will, like, where biology and engineering meet, and now you can have reusable components and kind of think of it as an operating system. If we were to go back to that concept and we zoom out now and think about the arc of biotechnology, not just CRISPR, but again, the convergence of biology, data, and engineering, which you're in all those. Where do you see this all taking us in 20 or 30 years? And what will that mean for society and healthcare as a whole?
Yeah, I think, yeah, especially if you're talking about a 20 or 30-year timeline, I think it's the classic people overestimate what you can accomplish in 2 years and underestimate what you can accomplish in 10. I really do believe that. And I think you're already seeing the hints of what I'm about to say in the liver. So in the liver, we're starting to see kind of the first readouts of clinical trials for a variety of diseases. And because CRISPR is this programmable technology, I think in the next like 10 years for sure, I don't see why there should be any patients that suffer from liver genetic diseases. Really don't see why we can't solve that problem. And I think then the next frontier would be how do we get to these other tissues like we talked about before, like muscle, brain, kidney, any cell in the body, right? And I think what you'll see in the liver is that it's like kind of slow and gradual at first, but then once the first therapy happens, it's followed by a rush of therapies,
Yeah.
and like it's kind of all like everything everywhere all at once. Once those first couple of therapies hit, and I think you'll see the same thing across all the different tissues of the body, basically, which means that in 20 or 30 years, I think you have a real shot at for diseases that have a known genetic cause we should really be able to potentially cure them, right? And live in a world where people don't have to suffer from known genetic diseases. Then the ball game becomes looking at, okay, what are things that we know are heritable, but we don't understand what causes them in the genome, and like kind of going into like maybe multifaceted diseases where multiple genes are causing something and doing multiplex editing and stuff like that. But I think it's really exciting that we can, with a straight face, I think, genuinely say in 20 or 30 years, no one should suffer from a monogenic genetic disease, period. Right? And that would transform the lives of millions. Even if we're talking about rare disease, the adage is very true that if you look across all the rare disease is not rare. We almost all know someone that has some kind of rare disease. So I think that we truly do have line of sight to that. And I think more generally, it creates this world where for the first time, we can actually say that, your genetics is not your destiny, right? Like you really, no matter what luck of the draw you got when you were born, you don't have to suffer from that for the rest of your life. And I think that very much could be, a major triumph of human engineering.
Yeah, it's an extraordinary vision. Trevor, congratulations on such wild success so far, and thank you for your time. You've been very generous with us. This has been a very insightful conversation for me. I very much enjoyed it, and I look forward to meeting you in person at the World Medical Innovation Forum next week.
Yeah, thanks for having me and looking forward to the forum.
That was Trevor Martin. Now, unnatural signals.
What stood out here wasn't ambition, it was restraint. Instead of waiting for a future delivery system, Mammoth designed biology to fit what already exists. Smaller tools, smarter fit, less waiting. That mindset changes everything. It suggests that progress doesn't require perfection, just compatibility. And once biology becomes something we can tune instead of endure, the idea of genetic destiny starts to loosen. The signal we're holding on to: engineering doesn't eliminate randomness, but it gives us a way to work around it.
Thanks for listening to Unnatural Selection. If you enjoyed the episode, subscribe, leave a review, and share it with someone shaping the future. Explore more at unnaturalselection.net and connect with us on LinkedIn and Facebook. Until next time, keep evolving.